Moving on to Andrew Yu from Washington University, Department of Developmental Biology. He's going to talk about modeling adult-onset neurodegenerative disorders by patient-derived, directly reprogrammed neurons. Andrew?
Thank you. Thanks for the having me here and participate. It's a very stimulating workshop. I changed my title a bit, so hope it's okay. The reason why I changed my title is because we have a slightly different way of generating human neurons, and this is a process that we call direct neuronal reprogramming. So what I mean by that is, as we do this human neuron degeneration, we have one thing that we keep in mind, is the notion that most neurodegenerative disorders are late onset disorders, and they also oftentimes they affect certain types of neurons. So today I'm going to be discussing about how we go about modeling Huntington's disease. For that we try to generate striatal neurons, medium spiny neurons. At the same time, we'd like to answer a few key questions. So if we can model the adult onset of the disease, how do we specifically and efficiently generate human neurons that are disease-relevant subtypes of neurons? And how do we use these neurons to study adult onset neuropathology of the disease? So my lab works on small RNA molecules called the microRNAs. These are small RNA molecules in about 22 base pairs. And we use two different microRNAs, miR9 and miR124. Developmentally, miR9 and miR124, these are two microRNAs that are upregulated at the onset of neurogenesis. And what we do then, we take those microRNAs, we topically introduce them into human somatic cells. So we use, oftentimes we use human skin fibroblasts. And what we try to do is we instruct the fate of starting fibroblasts to be turned into neurons. And this is why we call this direct fate reprogramming. And sorry, it's a bit of a busy slide, but the reason why we focus on these microRNAs is they have some properties that is very unique, which is the fact that if we look at target genes of these microRNAs, and all these genes that are labeled in red, they all share one thing common, which, although they do many different things, which is the fact that these are genes expressed basically everywhere, but selectively absent in neurons. So what these microRNAs are able to do is to recognize genetic network that exists in non-neural fates, and they try to erase it. And some of those genes are anti-neurogenic genes. So by the virtue of eliminating that anti-neurogenic property, then we can evoke a neuronal program. So what we do is we express microRNAs, miR9, miR124. We made them into one synthetic cluster of microRNAs. So from now, I'll just call them miR9-124. And we use them as fate reprogramming factors. What I mean by that is we topically express these microRNAs in human fibroblasts. So this is what the human fibroblasts look like. Oh, actually, I think. Okay, hopefully you can see. Okay. So this is what the human fibroblasts look like. And when we topically introduce microRNAs into human fibroblasts, this is what happens to those cells. And as you can see, now cells resemble neurons, neural morphologies, and they start expressing markers of neurons, including MAP2. And what's interesting about this process, we call this microRNA-induced neuronal reprogramming, is that if we look at like the fundamental aspect of this fate conversion, so we go down to the chromatin level. What happens is in response to this microRNAs being expressed in human adult fibroblasts, it induces the closure of chromatin state or chromatin accessibility that's important for keeping fibroblasts as fibroblasts. And after that, the chromatin opens up so that it's usually for neuronal genes so that they can be activated. So we call this microRNA-induced neuronal state, which happens in sequential pattern. So what I mean by that is from the starting fibroblast, microRNAs go in, they erase the fate first, Then after that, neuronal program kicks in. When the neuronal program kicks in, we can also combine that microRNA-induced neuronal state with different transcription factors expressed in different parts of the brain or in CNS. And by doing so, we can actually push this neuronal conversion into very distinct types of neurons. So using this analogy, what we've been being successful is we combine microRNA-induced neuronal state with a different set of transcription factors, in this case, as the CTIP2, DLX1 to MIT1L. So if we do that, then we sort of push them towards the striatal lineage. But we can also do the same thing. For example, if we combine with the ISL1 and LHX3, these are famous motor neuron transcription factors, we can basically push them towards motor neuron lineage. One very important aspect about this direct fate conversion is the fact that we can start with the fibroblasts that come from different individuals of different ages. And when we do this direct conversion, what happens is that the cellular age stored in starting fibroblast, basically that information carries over. So what we are trying to do is we try to borrow the age information from starting cells, but at the same time we alter the fate. So now they are neurons, but they think they're old. So this is one example using Steve Horvath DNA methylation-based epigenetic clock. So if we measure the clock before and after neuronal conversion coming from different individuals of different ages, we can tell them that the age clock basically stays consistent. Another demonstration that the age carries over, it comes from this study just got published a couple of weeks ago, where we look at the tau isoforms. So in this example, we look at tau isoforms, if they start from sort of fetal neurons versus adult neurons, then we try to compare what isoforms these neurons express. And developmentally speaking, during neural development in embryonic neurons, usually there's one isoform, it's called 0N3R. And in adults, we have 6 different isoforms, largely divided into 2 different tau repeat domain containing isoforms called 3R and 4R. And this red axon here corresponds to axon 10. Then the inclusion axon 10 basically defines tau to be 4R. And in adult brain, in healthy adult brain, there's a fine balance between 3R and 4R at the 1-to-1 ratio. And disturbance of that ratio actually can be pretty pathogenic. So for this, what we did was just briefly giving you the punchline from that paper is that when we do this fate conversion using microRNAs, and in this case, we are using one transcription factor called ISX9, the MIT1L, and we combine them with a small molecule called ISX9. We pushes them towards cortical lineage, and this is what the cells look like. And if you look at the amount of 3R versus 4R, and this is one example coming from mass spec analysis, they express 4R at the level that is basically indistinguishable from adult brain. And the point I'm trying to make here is these are neurons that are quite distinct from fetal neurons. And these are examples where we look at peptides that correspond to axon 10, and these fetal neurons basically don't have it. Right? So going back to what we tried to do with this microRNA-induced neuronal state for modeling Huntington's disease, what we do is we purposely collect fibroblast samples from adult-onset Huntington's disease patients containing the CAG repeat numbers in the range of 40, because we're trying to model adult-onset. And we get their fibroblasts, and we apply microRNAs and the CDM factors. So these are CTIP2, DLX1, 2, and MIT1L. And if we do that, then we get this type of conversion where we go from Huntington's disease fibroblasts now into medium spine neurons. So we collectively call them Hdmsns. And what's important about this directly reprogrammed neurons is the fact that if you look at some of the pathology that happens during adulthood, we can actually see some pathology. So this is one example where we try to look at aggregation of Huntington protein. We actually see them, and over time in culture, and this is an important point, over time in culture, when cells acquire the neuronal state as medium spine neurons, then we start seeing cells actually degenerating in culture. And this happens without adding any additional insult. So there's something about cell intrinsic property where these cells acquire the identity of medium spine neurons, then they start degenerating. And this is at the level of cell death. So knowing that, and also knowing the importance of age for adult-onset disorders, what Youngmi Oh, along with Songwon and Xiaowei in the lab, did was trying to see why we see this phenotype. So for Youngmi's study, what she did was she tried to collect fibroblast samples from patients before the onset of pathology, and also fibroblast samples from symptomatic patients. Then we turned them into neurons, and we tried to compare neurodegenerative pathology. So one thing that was striking for us is the fact that if we collect fibroblast samples from healthy individuals, but both young and old, and at the same time from patients, but before the onset of disease, and also compare them to symptomatic patients' samples, although the reprogramming is similar between different samples, what we see was pronounced neurodegeneration that only happens in post-onset neurons. And this is very important to us because we are trying to understand how neuronal aging contributes to the onset of Huntington's disease and neurodegeneration. And the major difference between pre-onset state versus post-onset state is aging. So knowing that, what we try to do is trying to compare what are the cell intrinsic differences between pre-onset state, post-onset state when they're turned into medium spine neurons from Huntington's patients, and also try to do similar things from healthy individuals. And we carried out this transcriptome analysis called weighted gene co-expression network analysis, WGCNA. And what that tells us is that when we compare pre-onset versus post-onset, there are certain pathways that emerge. So the one gene network module that emerges is called Brown module. And it's a module that is downregulated in post-onset cells. And we actually focus on this because this module contains a lot of terms enriched with the pathways and also phenotypes that we can actually see. And for example, apoptosis and cellular senescence. Also embedded in this Brown module is the regulation of autophagy. So why is that important at all for neurodegenerative disorders, at least in this modeling context? And so what we did was by going to post-onset medium spine neurons, then we started treating those cells with the inducers of autophagy. And when we do that, then we actually see cells being rescued from cell death. And this is one example. So we're using this compound called G2115. This is an autophagy inducer developed by Dean Perlmutter of WashU with his team. And when we do that, then we actually see cells being rescued in a kind of dose-dependent manner. And so the autophagy, the reduction of autophagy, and also we can try to fight it off by inducing autophagy pathways. So then the question becomes, if that's the case, then what is the cell intrinsic properties that are unique to symptomatic Hdmsns? And here's one example. We are talking about this one microRNA called miR29B3P. And this is actually a very interesting microRNA because with aging in the brain, this microRNA goes up a bit. But in the context of Huntington mutation, the upregulation of this microRNA is actually quite drastic. So it becomes exaggerated in terms of being upregulated. And this microRNA is important because it appears to be upstream of a lot of genes in the brown module. So by the same token, then if we inhibit this upregulation of this microRNA, then we can actually rescue cells undergoing neurodegeneration. So this is one example of what we can try to do by taking these patient samples and try to understand what underlies the onset of neurodegeneration. So with that, let me just summarize by saying that microRNAs, we use them as potent neurogenic molecules, and that we can co-express different types of transcription factors to push them into very distinct neuronal subtypes. And this is one example where we can try to understand how aging in medium aspiring neurons that may contribute to the onset of neurodegeneration. So with that, let me thank my lab members. It's very quickly went by, but it's a lot of work done by many different lab members, especially Young Mil and Sungwon Lee in terms of the modeling the neuro aging and Huntington's. And I'd like to thank my collaborators within WashU and outside WashU and my funding sources. Thank you.
Thank you, Andrew. For the sake of time, we're going to go straight into the roundtable discussion. So if you have a question for Andrew, Steve, or Virginia.
So I didn't realize you could do reprogramming just by microRNAs. It's amazing. What is the yield like? Or you don't need any chromatin modifying agents or anything in general to get this to work? I'm just maybe just explain a little bit more how general this idea is.
Yeah. So it's an idea that basically started when I was a postdoc in Jerry Krapschu's lab. So in terms of the microRNAs alone, luckily for the purpose of reprogramming, because they target a lot of chromatin modifiers, and at the same time, what they also do is they instruct chromatin remodeling complex to acquire neuronal function, because it instructs the chromatin remodeling complex to swap out their subunits. And I think
the sort of degree of whatever localization of the chromatin modeling agents that is specialized for neurons. Is that, I mean,
yeah.
Normally you think of them as being, sort of genomic level and operating everywhere. So I would expect it could wreak havoc, but it might not be that specific, but I guess it is.
Yeah, so one example is like some of the subunits, they're very specific for neurons. So I say microRNA-induced neurons, but what's more important is understanding the importance of chromatin state that determines the cell fate, and that's what this microRNA is trying to mimic. So it's really chromatin rich. Yeah. Steve.
Thank you. Yes, thank you for the very nice talk. I have a question that is more kind of a clarification about what you mean with directed in particular. And so do you mean the kind of directed reprogramming in the sense that is kind of in the operational sense, so that the protocol is kind of one step, or phenomenological? Because in many of, I've been doing a lot of, for example, single cell transcriptomics of many protocols, and I've never seen any protocol where the actual phenomenology is directed, like in one step. It always passes through neural progenitors, neuroblasts, and neurons. So could you comment about that?
Yeah, so like what defines direct? So, I mean, we did a lot of single cell and like all the genomics, ataxic and all of that. And in terms of how cells behave, I mean, the most important part, the reason why we call it direct is the first thing that happens is that the cells when they start expressing these microRNAs, they come out of cell cycle as fibroblasts. And over time, then they become neurons. Then when we look into changes in chromatin state, what we saw was all these regions that define cells to be non-neuronal fate of fibroblasts, those are the ones that close down. And over time, then after that happens, and the reason why we call them sequential activation is because after that, the neuronal program kicks in, and all this neuronal loci opens up. And this happens like without without going through this the kind of progenitor or cell stem cell, not cell, the neural stem cell like proliferated state, and that's why we call direct conversion, because it's actually going from post-mitotic state, one state to another. Thank you.
Steve?
Hey, Andrew, great talk.
Thanks.
So I know I've kind of asked you this before, but maybe if you could just talk again a little bit about when you do the direct conversion, if you do like a UMAP on the transcriptomics, does it completely look like a neuron, or does it have any vestiges of a fibroblast? Is sort of one question. And then kind of related to that is, what are your, like just conceptually at like a higher level, you conserve the epigenetic marks of aging, but dramatically change the epigenetic marks associated with cell type. Where do the other epigenetic marks fall, like environmental influences or things like that? What gets retained, what gets saved? What are your ideas about how that works? And then one more question. Senescence. I wanted to know, so fibroblasts, they undergo senescence, right? So how is senescence and aging, and what happens if the fibroblast is senescent? Will that work?
How many questions was that? Okay, the first one. Let me try to go backwards from the senescence. We actually, in terms of causing cells to senesce, it requires us to passage them multiple times. And then really age or make starting fibroblasts to senesce by multiple passages. And we can't actually do that experiment, because once the primary fibroblasts, when they senesce, the reprogrammability actually goes down. So it's a bit of hard experiment that we can do. What we can do is we can go to non-senescent cells, make them to be post-mitotic, then induce neuronal reprogramming. But in terms of the senescence, it's kind of hard for to do the experiment. The second question was epigenome, right? If we look at chromatin accessibility and DNA methylation patterns before and after conversion, we actually do see changes with other loci and the methylation patterns of other loci. So we do see that, and also we see chromatin accessibility, quite drastic changes, but not the epigenetic clock. So the way I'm thinking about it is there's got to be, because it's it's a DNA methylation transferases, there's got to be those enzymes, but there's got to be cofactor that senses the age, because otherwise we just it's a black box for us. We don't know. Actually, we are actually trying very hard to find that.
It's an impressive black box.
Thank you.
Any final questions? If not, for the sake of time, I think we will move on with Rohit.